Thank you very much, Mark. And thank you very much all for coming. So, we're going to talk about something that maybe many of you have an intuition of, and I'm going to try to convey how we tackle the problem of, a little bit body language and body posture over time, and how we grow when we are like, kids or teenagers, and how do we maintain our straight body throughout time. And it's a little bit of a challenge. So you've probably seen that with time we tend to, change our posture, and what we start to understand is that actually there is a physics of like how to keep a straight body and to maintain a straight body. And we discover the sensory system inside our bodies that enable to detect how straight we are and to adjust if we have like a bad posture. The sensory system is very redundant and robust so that we can keep the body straight, because it's critical for living well and surviving well in this world. But you're going to see through the talk, it's actually also responding to other kind of injuries or attack, including pathogen if you get a viral or bacterial infection. And so my talk is really the beginning of us understanding how we grow a straight body and how we keep throughout life a straight body. And you're going to see that it opens a lot of questions that we haven't solved yet. We start from animals and we're going gradually into humans. So if I get to see you next time, I hopefully can tell you what's going on in the human body. Okay? So this is like the work of actually many people who, because I'm French and American, I actually have a team that is very much French and American. And this was like the first version of the team, and then it evolved and evolved and evolved. We started almost 15 years ago next year. And every time half French, half American, and we keep this, balance quite steady because we love just interacting with, different programs, including KITP. That is really critical for us to bring physics and biology to understand problems about sensory integration and motor control. So we're going to start with a little game, okay, just to get started. And I'm very much encouraging the young people, as the others, to answer my questions here. But you have not learned at school what is, body language, but you maybe have a feel about what, different posture tell you about the state of a person. And so if I show you these people here, we're probably going to a music concert. What do you think their body tells you? Like how do they feel? What do you think? Excited, outgoing, energetic, right? Sorry?
Defiant.
Yeah, defiant. And what about this person here? What do you think? Sick? Depressed? Down? Yeah. And what about this little girl here? Yeah.
Happy and playful.
Yeah. And now, how about this one? That's a problem, right? Especially for those guys and for all of us, right? So this one, it looks a little bit like sick, but it's just a very bad posture due to the smartphone, right? And so this one to finish.
Joy.
Yeah, joy, energy, a lot of energy. And so in this posture, we have like, a very, kind of short time scale where we're just controlling posture and we're adjusting posture. If we're walking on a slippery surface, we have to adjust. So we do that on a short notice. But then you may have noticed that your body change also posture with time, and that you don't stand the same way that you maybe stood the year before. And sometimes if you have a very bad back pain, you're going to start to, to change your posture, and it's going to be very hard to recover. So there are long-lasting change of the spine that actually are occurring throughout life, So in humans, we are bipedal, obviously we stand on 2 feet, and we have a really beautiful, shape of the spine where you have a curvature on the rostral end and a curvature on the caudal end. So at the thoracic and at the lumbar level. And this, spine is really considered throughout vertebrates. The spine itself is made of vertebra that surround the spinal cord, where you have all the neurons that are critical to both sense, all the information that you get from touch, for instance, or if you burn yourself, or if on the contrary you're very cold. In addition, the spinal cord has all the neurons that enable you to walk and stand. So it's really critical, the spinal cord, to protect, because without spinal cord, you cannot stand, you cannot walk. So the spine is protected by, the spinal cord is protected by the spine, and many, in fact, deformation occur because the spine is critical to keep the edifice, the body straight and rising. But you can have, typically in men, Sherman disease that is called kyphosis, where with time you get a little bit of the curvature of the thoracic part in the ventral region, like ventrally directed. And you also can have lordosis, where you have a high curvature of the lumbar spine. And all of those are damaging your disc, and they, generate pain often, but they are also, a little bit of a challenge when you're like doing physical activity. And there is something that happened mainly at the age of you guys in the back of the room. So scoliosis is like a different deformation where now, you're going to have not a 2D deformation in the sagittal plane where you only go like this or you bend, like that. You actually get a 3D deformation of the spine where it does a little bit like a corkscrew, where the spine will now start to bend in 3 dimensions. And this happens in 3% of the population. So it happens a lot actually at the teenager age when you suddenly start to grow very fast. And when you grow very fast, at the moment where you can have those deformations occurring, and the problem is that you're going to keep them, and they can actually, usually if you do nothing, only get worse. So it's really a critical time where you're typically checked every year for scoliosis at school. But it can also happen with age. And so many patients with Parkinson's disease also develop scoliosis, which is more like of a late onset scoliosis. And then you maybe have seen individuals who actually get this condition, which is like called campocormia, where you have, when you're standing, a very difficult time rising yourself up. And so instead of pushing on your feet like we all do without thinking to stand straight, patients who have campocormia cannot really, when they stand, raise vertical their spine. And it's even worse when they're walking. So sometimes, I don't know if you've seen people who are suffering from that, but you have a straight body, but when you start walking, it goes back to this, and then you can straighten it again when you're walking. And it's quite of a fascinating condition, because if you take a patient who was in La Pitié-Salpêtrière, the hospital my institute is affiliated to, and you lay them on a bed, you see that their body is completely fine, stretching out and being horizontal. It's only when suddenly they are, on their feet that they can't rise vertical. And when they start to have this imbalance of going out of balance to walk, then it makes it even worse. So they are like almost forming a 90 degrees in the worst cases. So I'm going to do a little bit of a basic, neuroscience to just get everyone on the same page on what we understand in neuroscience. So we know that this condition, like scoliosis, for instance, a 3D deformation, they are very much dependent on neural information. And people, doctors I've talked to before, when I started my team, told me there must be gravoceptor in the body that enable you to tell what gravity, where gravity is. It's not in the head, like the vestibular system that tell you if your head is, vertical or tilted. And there must be some gravireceptor in the body that tell you that your body is vertical. And they knew this because they had evidence that the posture of people with scoliosis was affected, that their reflexes were not correct, that they were not reacting properly to sensory motor reflex, and so there was a neural component to it. It's not congenital, it's really like coming with age when you grow very fast. And so in neuroscience, we understand that sensory information from both the world, so exteroception, and also from within, interoception, are really important for adjusting not only our immediate response to an information that we come across, but also to adjust our motor patterns in terms of locomotion and posture. And so the posture really rely on a lot of sensory information. And then based on this change of posture or the fact that we're moving, we have also a feedback that comes from the motor action. And at every level, from the muscle to the spinal cord or the spinal cord to the brain, you have always feedback. It's really important. The feedback is critical for adjustment of your posture and also for speed, from what we tested in experiments. And then in addition to that, you have those internal states. So if you're sick, you're not going to move very much, and if you have back pain, you're going to change your posture because a lot of the muscle and inflammation will change a lot the activity of muscles around your spine. And then you also have effect of emotions, of stress, and many other parameters like that. So we have like an effect where sensory inputs can directly change your internal states. One example for us in Paris is, you go to work and you have a lot of people like just insulting you on the way when you're biking, or if you're in the ground transportation, you come to work and you just feel a little bit different. But you also have like change of internal state that can, come from infections, or external parameters that you don't control. Like, in Santa Barbara, you have a lot of light, but for us in France and many people coming from the northern of Europe, we have like phases where we don't see the light very much, and those change a lot your internal state actually. And so you have from this, transient, very fast acting sensory motor loop, you have also long-lasting effects that can change a lot your posture and your internal states. And so if you look at the textbook, how do we control our posture? What you're going to find at this stage still now is only really one word, which is called proprioception, which is like this integration of the body position that we use both for posture and for movement. And the information that people have studied in animals, Sherrington was studying it in the cats, but it's been looked at in many animals since, is dissecting the spinal cord that looks beautiful like this, like a butterfly in the cat. They saw that there were receptor neurons that were outside of the spinal cord. They are peripheral. They're not in the central nervous system. And they had their axon either under the skin to detect, touch or heat or cold, or they could also have axons in the muscle itself or on the tendon, and they could sense the tension of the muscle or the tendon. And so most of our understanding is really based on the work of Charles Sherrington, who showed that this receptor neuron, they can act by modulating the motor neuron directly in the spinal cord by sending a very long axon that goes to the motor neuron, which then project on the muscle and contract muscle. Or they could also go via another neuron that we call an interneuron to modulate the motor neuron. And typically the Babinski reflex, when you kick here and you see your knee moving like that, is actually coming from those reflexes. And so the textbooks are still focusing only on posture being controlled by the spinal reflex in order to change our locomotion and also to change our posture. But when I was starting my team, I thought, well, it's a little bit strange that you would use only the information from the periphery to know that your body is actually straight. Because when you think about it, for us, it's only 2 feet that we can collect the information from, we push against our feet, the sensors in our feet are able to send information back to the spinal cord, but it's very little information. It's information that goes from here to the lumbar region, and I have no information on my thorax or my cervical with those 2 feet. So I thought, well, maybe we're missing something. And so when I looked in detail, at that time I was in UC Berkeley, I actually came across a super funny cell that look a little bit like a Perrier bottle. They have like a pear shape, and they are both ventral, left and right. And these neurons look like hair cells from your ears. That's the one that you use to listen, to hear sound, except that their apical extension is a little bit disorganized, and they have an axon that can communicate with other neurons in the spinal cord and in the brain. So they have the way to communicate with the motor circuits all across the brain and spinal cord. So these neurons were found by 2 anatomists, Kolmer and Agler, who were working on this already more than a century ago. And they described when they were doing those experiments in farms and zoos during the First World War. So they would collect dogs and camels or cows and pigs, and they would dissect the spinal cord, and they labeled them with a Golgi staining to see them black like this. And then they would print, the images they see. And both of them discovering these cells said they have to be a sensory system. And we think that they probably constitute a 3rd ear in the spinal cord. So we have 2 ears, but we would have one that senses something mechanical in the spinal cord. And so I discovered those papers when I was starting my lab, and I was really excited. It was written in German, translated it the best I could. And then I read that they had a big fight. Even though there were 2 people in the world working on this, it's really good. They had a big fight about what this sensory system was doing. Kolmer was convinced that the ciliated neurons were interacting with this black dot here that is called the Reisner fiber, a very long polymer I'm going to show you in the next slide. And Agler said it's an artifact of fixation, this is nonsense, and so they were writing in their books really strong debate about, the fact that the other one is wrong. And so to explain you the Reisner fiber, I need to go to 1860. So Ernst Reisner didn't look very happy on the picture, but he is the guy who made it happen for anatomy in the nervous system.
His posture is good.
Yeah, his posture is, he didn't have a smartphone, so no computer either. And so Ernst Reisner described in a paper in 1860, when he looked under the binocular at the spinal cord of lamprey, he saw that there was this big, big polymer inside the central canal, inside this, conduct, like a canal inside the spinal cord that lets the fluid from the cerebrospinal fluid from the brain flows. And he described that there was a polymer there that he could see just under the microscope. And then after he did this, we looked with electronic microscopy, and we saw that indeed we can find it in the rat, in the cow, in the lamprey, like he did in the trout and in the frog. Every species will have a different size of the polymer, but the remarkable property is that the monomer is highly conserved. So the unit that enables to form by aggregation a polymer is this gigantic protein called osteospondin that has one domain in red for aggregation and then many domains for interaction with itself or with other partners as well. And so it was such a big, big protein that nobody had attempted to try to knock out the gene and see what it does. So when I started my lab, I got really excited that we could actually take a look at this. But people told me, oh, you're going to just look at those ciliated neurons that you find in one species. Are they really conserved? And so I had read the paper in German with the help of my first grad student, Osbaum, who was German. And so I knew that they were conserved, but I had to demonstrate to them because I don't read the paper in German. So what we did is that we looked for these cells in macaques, in mice, and in zebrafish. Every time an animal would be sacrificed or euthanized because it was very old or so on, we would actually come and we therefore dissected 3 spinal cord of macaque that were big like this. It was a lot of work, was very interesting. And we found that those cells were very dense all along in the cervical, thoracic, and lumbar region in the macaque. We find them also in the mice, and we find them in zebrafish. And so they're always ciliated neurons that are inhibitory neurons, which is very unusual for a sensory cell. And they have a receptor that has, an acronym that is not interesting to remember, but a receptor had been found in your taste bud, in the tongue, to be important for sour tasting, when you're tasting like, sour in the mouth. And so we decided to study this question in a fish. And I'm going to explain why. Because you see, it's very hard to go inside the spinal cord of a human. It's not transparent, and it's very big, and it's very invasive. That's one of the major problems of studying humans to understand the spinal, those gravoceptors that could be there, is that it's very hard to get into the spine. It's very invasive. And so we use those little animals because they are only 4 millimeters long, and they are vertebrates, so they have a lot of conservation. And they are really smart in the sense that at 4 days, they can already hunt for prey and find food, which we managed to do much, much later in humans. And then what is very neat about this animal is that because the animal has a transparent body and transparent brain, everything is transparent at the larval stage, we can actually look at how it hunts. For instance, in this movie, we took with a little paramecium that the animal would like to eat. We put the animal in an agar bridge where it forms a V shape like that, so it cannot go anywhere. But it continues to track with its eyes the prey, and it continues to move its tail to try to, eat the prey when the little paramecia becomes close to it. And so we have a way to do what is the equivalent of functional MRI in the human brain, but with a single cell resolution. So we can really go at understanding how the nervous circuits work. And we can look at the structure of neurons, so we can label neurons with different colors. That's a technique called rainbow, where you can see each neuron at the cellular resolution. You can even see their connection and their synapse in the brain. And then you can also use sensor for activity of the neurons. And here is a sensor called G-count, where we can look at throughout the brain using infrared light to go deep in the brain with 2-photon imaging. And then we can see the activity of each of these donuts is one neuron. We can dive in the entire brain, and we can take a look closely at how the neurons interact with each other by just looking at those, cells together. So it's very beautiful. I want to share that because I did my PhD in the lab of doing a lot of optics for fast imaging. Sorry?
How is that produced?
So this is like a 2-photon microscope with a resolution about half a micron. And then we can go deep in the brain. We can reach the very, very most ventral parts, close to the mouth, that is 500 micron deep. And we just, line scan to get like the activity of the brain at typically 4 hertz. So we can look at activity of the brain at 4 hertz. And so people have used different kind of microscopy. When I was doing my postdoc in Berkeley, Jan Huiskin was developing like light sheet microscopy, and with light sheet microscopy you can go even faster, so you can go 100 hertz to look at the activity of the brain, and therefore you have a way in the little fish brain to look at activity of the entire brain when the animal is performing a task, so either hunting for a prey or looking at visual stimuli or mechanical stimuli and so on. So we have this power that we can image the entire brain at the single cell level, but we also have the advantage that the model is very cheap and it grows very fast. So in 5 days, you already have like a brain that can do a lot of processing, and therefore you can do experiments quite fast. When you're impatient, it's very useful. So I'm very impatient. I'm working on it. And so we have a really good model to like investigate what's, the system of those gravoceptors could be from the prediction of the medical doctor.
Which direction am I looking relative to the body of the fish?
Yeah, so here it's actually a dorsal view. So it would be like now it would be an image where I'm looking at an image of, what will be a frontal plane in humans. So I'm just looking at the dorsal view. So it would be like looking at the back of your brain from behind. And so here you have like the brainstem, or you have a lot of the motor commands, and here you would have the cerebellum, for instance. So now we want to go back to this sensory question of how do we know that our spine is straight, and how do we grow a straight body axis and maintain it straight. And for that, the zebrafish is great because we make those transgenic lines where we can use genetics and CRISPR to, you know, like make transgenic animals where only one cell type is going to be fluorescent and show us the activity of those cells. And here you see the cell that I told you about, the CSF contacting neuron. They're all along the central canal. They form really like a line, you know, in the body. And if we zoom a little closer and we look at one of those neurons, they really form what Colmer and Eider had described, the little, like, you know, Perrier border with the apical extension in the central canal where the fluids of cerebrospinal fluid that bath the entire central nervous system is going. And then they have an axon that is going towards the brain. And so Ursbaum, the first, student to tackle this question, did experiments where we have the spinal cord with those cells, you know, on the left, on the right, and in the ventral side. And Urs tested how they respond to mechanical compression when we are bending the tail, either, you know, ourselves, or when we actually trigger a little response called an escape response in the fish, where we puff in the otic vesicle on the right, and we get the animal to respond by a large cell bend on the left to escape from the danger, the stimulus on the right. And so we do the same in human. It's called a starter response, where you're going to like try to, if you have a big sound, go away from it and close your eyes to protect yourself. And so the fish does it very nicely. And when Urs did those experiments, he discovered that only the cells that were on the left side, dorsal and left, were responding to the left bend when the compression occurs on the left. And on the contrary, when he tested the right side, he got, you know, the right cells to respond to compression and not the left. And if he tested now a ventral compression, when he pushed and made the fish do like abs equivalent, you know, like that, he actually got them to respond only on the side of compression. And so we thought at first, okay, let's look at the flow of cerebrospinal fluid that is going in this cavity. And we use little beads, fluorescent beads. We also use a technology that is called optical current tomography, where you can look at endogenous particles in the canal, and you can see their movements, the same that you can do with fluorescent bead. And we built model of the flow, and at first we thought, okay, let's look at the flow when the animal is contracting its muscles. And what we saw is that when the contraction occurs like now, the flow is immediately very strong in the direction of the contraction of the muscle. So when a fish, you know, swim in the water, if you look at fish swimming in the water, they have to do an ondulation of the tail that is actually a wave coming from their head to their tail of activity in their spinal cord. And while the muscle contracts from head to tail, the flow in the CSF is very fast flushing from head to tail. So I don't know if we have any yogi in the room, but if you do ocean breaths when you're doing yoga, you know, if you have this very deep, like breathing, you actually will have the same in the cerebrospinal fluid around your vertebra. You will get the CSF to go up with your inhale and to go down with your exhale. And nobody has looked inside the spinal cord in humans because it's very hard to do this experiment. But it seems to be the same. It's like a peristaltic pump. And so at first we thought, okay, maybe the problem is solved simply by the fact that the little neurons here expressing the activity sensor are just activated every time there is a muscle contraction, because it flows, you know, it gets a flush of the cerebrospinal fluid and they get activated by the flow. So we thought, you know, maybe these neurons are just a flow sensor inside the body. And that was our first idea. And then we talked a little bit more with experts of fluid mechanics. And when we discussed with them, in particular François Galère from EPFL and people from KITP last time I came, we realized that, you know, it was not a very good way to sense. And it was very hard to explain that you could sense left, right, or ventral with just a flow sensor, because the flow is null at the wall of the central canal of this cavity. It's maximum in the center, and when you're going to have the contraction going or the bend happening on one side, you will get a small deflection, but this is only 8 microns, and it's going to be very, very small, what you get, probably under a micron. And so very hard to imagine that it could really come from flow only. So we thought a little bit more about the debate of Kolmer and Alder together, and we thought, well, could it be that this fiber found by Reisner in the 19th century could actually bring the asymmetry and could be the solution? And so we did with Pierre-Luc Bardet, a geneticist in the lab, we made a knockout where we come with the little scissors from CRISPR, we cut the genome right at the beginning of the first exon in red here, and then when we cut the genome, the machinery to repair is very active during development. You don't want any mutation that would create a monster. So the machinery comes to repair DNA. And when it repairs the DNA, in one case, it put a stop codon, so it just stops the reading of the genome. And in another case, it added 15 nucleotides, adding just 5 amino acids to this gigantic protein that forms the polymer by agglomeration. And when we did that, it was really cool because we took an antibody that we raised against the cow Reisner fiber, and we could detect the fish Reisner fiber because it's so conserved. And we saw that the fiber was there in the wild type, but in the mutants, when we did a null allele or when we just added nucleotides to change, add amino acid in the aggregation domain, we also had no fiber produced. So the fiber cannot aggregate. There is no fiber in the body. And we saw two things happening. So the first thing we saw is that when we don't have the fiber, the sensory neuron that respond to the left-right bend could not respond anymore to left, right, or ventral bend. So the sensory system detecting curvature was broken. And we thought, okay, what could be explaining this? What could be a polymer in the body, used for? And we took a close look at the fiber with a collaborator in the US called Ryan Gray, who made, also again using the CRISPR technology of the scissors, he added a little green fluorescent protein to the fiber. And so the fiber became green in vivo. So we start to look at it across, the body of the animal. And when he did that, we saw that the fiber was like a straight line, like Reisner described it a century and a half ago. But we also saw, I don't know if you can see it, that it seems to be active. Do you see that? It seems to be moving. It's a little strange. It's not very big, the amplitude of the movement. But you see the fiber is 3 millimeters long. We're just looking at 200 micron here. And we can see little fluctuation. We thought, are we crazy? Are we not crazy? And we fix the animal in formal. And when we do that, after fixation, we don't see any movement. But we really see it only when the animal is alive. There are lots of fluctuation of the fiber. And so we ran an analysis to understand what were the main mode of movement of the fiber. We saw the main mode is a translation, where it goes up and down in the body, ventral dorsal. And the secondary mode was a rotation, where it forms like, a little rotation across like that. And then with a student from UC Berkeley, Céline Bellagardin, who came to the lab for 1 year, we looked at this fiber a little closely, and we saw that when the fiber moves, the neurons that are sensitive to curvature are spontaneously activated by the movement of the fiber. If we cut the fiber, we lose their activity. And when we label now, also in green, their cilia, we saw that their cilia are like beating, and they're touching and going touch and go with the fiber. So they can like touch the tip, of the fiber. They can stay attached for quite some time, and they continue to beat, but they touch the fiber. And so with my dear mother, who is a physicist, so it's also a story of physicists. My mother has done a lot with Phil Pincus here in UCSB, actually working on multiple aspects, but she's been really interested in cancer progression lately. She had started her career as a PhD student working on polymers. And so I came to her and I asked her, we never published together, but I asked her, could we try to model this fiber together, because you worked on polymers of DNA in the early stage of your career. And she was very excited to do that together. She's 80 years old this year, so it was really cool that we got this paper published on her birthday. And we did experiments to test her ideas that we have the fiber here you see labeled with the GFP. And then we have the little neuron sensory that are like those donuts you see all across. And we came with Céline and did experiments where we come now with a pulse laser and we cut the fiber inside the body and we look at how it retracts in the body. And when we did that, because it was so straight, we were curious to see if it would be under tension. And what we saw is that it was reacting like a spring under tension, retracting like that. I'm slowing it down 10 times to show it to you. So it's like straight at the beginning. It's also moving and active. And as soon as we bring the laser, it starts to retract like a snake, you know. And so my mother was really excited by that because she's done models on the polymer of DNA similar to that before. And so she told us, you guys have to plot the retraction distance as a function of the square root of time, because if the fiber is a little bit like, you know, a string that would be under tension, it would like, you know, have equal force of pulling and friction when it cuts. And then you see that the retraction distance should actually go as a square root of time. And she was right. And so we plotted, always good to check on your parents sometimes. So we plot the retraction, you know, distance as a function of square root of time. And finally, we got this beautiful trace where, you know, we did these experiments on over 300 animals with Céline. And we saw that you have very nice, you know, straight lines that, you know, give you the diffusion coefficient of the fiber. And then we saw those things of sticking to cilia, where suddenly the fiber attach and it sticks and it doesn't move anymore. And sometimes it sticks and then suddenly it jumps and it sticks again. And so we saw that it was very sticky material, this fiber, and it had like interestingly different tension in the neck than in the lumbar region. So in the neck, in the rostral part, it was very high slope, very high you know, retraction speed, and then in the caudal region, more like the, you know, to be lumbar for us, it was very low. So a very interesting property of a straight, you know, polymer under tension with heterogeneous tension that is much higher in the neck than it would be in the lumbar region. So we have this model where when the fiber is like a rope under tension or a guitar string, you know, except that you're bending the guitar. And so what happens is that when you bend the guitar to one side, like for instance the right side, you actually get the cell on that side with the little cilia to detect the contact and to respond to it, because you have this, you know, touch and go, you know, natural affinity for the cilia onto the fiber. And we are now investigating a little bit in more detail what is happening here and what are the receptors that we're finding. Now we find that this sensory system is really important for posture. So if we look at an animal losing, you know, like losing this sensory system, we find that the animal lose posture, and so they start to roll and lose their balance when they are like swimming. And so it's very important for posture. And it's not only important for posture, but it's also important for locomotion. So we use optogenetics, where you activate the cell and you look at the response with electrophysiology on the target. We used a technique I had contributed to develop as a pioneer in UC Berkeley, where it's called optogenetics, where you can activate neurons with light. So we use different opsins that come from chlamydomonas, for instance, where you can bring light on the receptor to activate a channel and let the ion go through. And we did that. We expressed this opsin from chlamydomonas in those little cells to activate them and look at their connection on the spinal circuit and in the brain. And so it's a diagram of mapping connectivity. It's not very important, the detail, but the importance is that we understand how they manage to control the posture and avoid the torque to happen so that the animal doesn't lose balance. And we also understand how they are able to, you know, speed the propagation of the excitation to speed the locomotion speed of the animal. And where we got excited is that what we found for the fish is also true for the mouse, where people have found that if you lose the sensory system by killing the cell in the mouse, you will see that the mouse tends to, when the mouse is challenged on a ladder like this, when it's losing the sensory system, it tends to fall, you see? Tend to do all these movements where it's not going to be very accurate and it's going to miss the ladder. And that's really interesting because it looks like the sensory system is conserved in mammals. And so, oops. Now, what is really exciting is that it doesn't control only how you change your posture on a short time scale. It also changed the way the body axis grows. And so we saw that when you lose the fiber, the animal is not able to grow straight, and they grow immediately curl down. When you lose the receptor I told you about that they're expressing, you get the equivalent of Sherman disease, where the fish will grow a spine that looks like a hunchback. And very interestingly, it's mainly there in men for humans, and it's mainly there in males for fish. So it seems to be very conserved. And it's more also strong, the deformation in males, both for humans and for fish. And similarly, the scoliotic phenotype that we get also when we lose the fiber at the adult stage, the torsion of the spine, you can see here that starts at the juvenile age, like in humans, is also much more severe for female, like it is also more severe for female in humans. So without, I think I'm reaching my time. I don't want to go into detail, but we have found like different peptides that can go expressed by those neurons into your cerebrospinal fluid, and then they're transported to the muscle, and they can act on dorsal musculature to compensate for you to not, curl down on the ventral side, and keep the dorsal musculature tonically activated to keep this beautiful posture. So that's what we found. And then just to finish and open on, the question of pathogen and how they change not only our brain activity with the brain fog of COVID, but also our posture, we also found that the sensory system was responding to attacks, including bacteria and virus, that enter into your brain and manage, for instance, when you have a cut, to use those dorsal root ganglia neuron I showed you in the beginning to actually enter into your central nervous system. And there they can attack and potentially change also your posture. And so I'm going to show you those evidence. So the fluid is all over the brain and in the spinal cord in the central canal. It's a very easy way for bacteria or virus to actually proliferate and get, for the bacteria in your nervous system throughout. And so that's why you can get meningitis, where a bacteria enters into the cerebrospinal fluid, multiplies in the fluid, and then can destroy your motor circuits and lead to paralysis. And so with Andrew Prendergast, another American fellow from the lab who is now in Yale, we've been looking at this question, and we found using molecular techniques to look at expression of this ciliated neuron that they were expressing a lot of taste receptor and receptor that enable them to taste metabolite from bacteria or sign of injuries. When you have an injury, mechanical or from virus killing your cells, there will be a lot of glutamate, ATP, RNAs all around, and these cells have the receptor to detect it. So we've done this crazy experiment during COVID with Andy, where we went to a lab of microbiology in Amsterdam. We used their facility where they had Streptococcus pneumoniae, which is a pneumocoque that is like the major source of meningitis and bacterial meningitis in humans. It's responsible for 40% of meningitis in humans. And they had it infused with a red protein, so we could inject the bacteria in the brain of the zebrafish larva and look at it propagating all in the central canal. And when we did that, we saw that our little neurons were becoming very active. So they started to show this very strong pattern where a cell is going to stay active for very, very long. And we demonstrated that if we remove those neurons again, we were actually reducing a lot the survival of the animal. So they use this sensory system to detect that the pathogen is there, and then to, in fact, secrete a lot of peptide that will kill the pathogen directly, or that can act to, boost the immune response. And so one thing that we noticed is that when we looked at the infected animals, their posture changed. So after a while, they would start to show dorsal arching. And we were really interested because that's also what we get when we activate the neurons. We also get the fish to show this dorsal arching. So they have, the opposite of losing the fiber, that is ventral arching. And if you look at the painting from the 18th century, you see that people who were not treated for meningitis would start to show this phenotype where if you didn't have at that time antibiotics to kill the bacteria, you would actually have an infection where your body goes into opistotonos, it's called, where you show dorsal arching, that if you reverse gravity, looks very much like the zebrafish larva with a dorsal arching. And so we have found a very interesting system where you have in the center of the spine, in the spinal cord itself, a fiber that is a very long polymer acting with ciliated neurons in order to change, posture and locomotion by detecting mechanosensory information as well as chemosensory information, like tasting the presence of a pathogen. And it can modulate not only posture and morphogenesis, but also innate immunity and help to face spinal cord injury. So we've done a lot of this by interaction with people in KITP. And in particular, we're very excited now to take it to humans to start to look at human tissue, to find the system, and to look at mutation we find in patients with scoliosis. And we see that the fish, when they have the mutation, grow also 3D deformation of the spine. And we're at a stage now where we're finally accumulating enough evidence to work with theorists who are interested in morphogenesis, in KITP, and to try to model this to understand, how it works at the early stage of development and how it managed to keep, the straightness of the axis throughout life, when, in particular, during aging. So thank you very much. I hope I was not too long.
We have questions. I'll ask one. Is that fiber you showed us is free at one end?
No, it's actually attached on the two ends. Yeah, it's attached on the two ends. And then we are very curious to work with Boris Sherman here. We started to discuss a lot with him and his postdoc and Sebastian Streschen about okay, so if it's attached at the two ends, how do you keep the fiber under tension? So we think that the cilia play a role, but we think also that there is shearing between the different tissue, and that the spinal cords and the different epithelia are actually enabling to keep the fiber under tension. So we're going to do more experiments to test that idea. Yeah.
Yes, John.
So as you age, you usually get smaller.
Yes.
2 inches smaller than So is it not because your spine going on its path is any different length total, but that it's scrunched down because of the curvature, because of the things that you've been saying?
So there is, I think, a combination of multiple effects. You have often curvature occurring on later stage. So one stage is teenager time where you grow very fast, and you can have those defects that can really lead to scoliosis at the teenager age. But then it's true that many people observe that you also have much less control of the straightening with age, and you can often get Sherman's disease like that in male, and the scoliosis that is really 3D deformation that you can also observe. So that will clearly reduce your height, and there is also I think the properties of the disc. Don't worry. Properties of the disc also are also changing. So I think you also lose on the space in between each vertebra, I think. So both are acting together. Yeah. So I think I think but it's there are lots of exercise that you really need to do to actually defeat aging. So the very important exercise is to really push a lot. So you push, when you're standing, but you also need to do those exercises where you push on your forelimbs and then try to do as much as possible the stretching. Because those sensory motor loops, they are very plastic throughout your lifetime. So you need to kind of force yourself to not stay too much on the computer, not watch too much, TV, and have those moments where you both are going to work on the strengthening of the deep muscle, but also on the stretching your body, because that flexibility is critical actually to keep your system as long as possible. So elongating the spine It's really critical. Yeah.
Wait,
Yes, but
spinal cord injuries, any comments?
Yeah, so I didn't talk about that because it's in progress in our lab. But in fact, we were really excited about this because we saw that the same machinery to detect that pathogen are coming is also used to detect that there is an injury in the spinal cord. And so we have evidence that during an injury, the content of the cells is going to be released in the cerebrospinal fluid, and these cells can detect there is an injury, and then they act via the release of a neurotransmitter called GABA. They can act on the stem cells, and they can change regeneration in the spinal cord. And so that's a very interesting place where we'd like to know, in fish, it's like salamander, you can regenerate the spinal cord. So if you do an injury, you can recover really well, and the animal doesn't stay injured and immotile like we have in humans. And so one of the questions that we wonder is whether in humans, this sensory system is acting differently or might be, inhibited in the way that it's operating in fish. Because in fish, they're very important for detecting the injury and recovering from the injury to enable the stem cells to regenerate the spinal cord. So we have actually Julius, who got the Nobel Prize in UCSF. He actually published a paper in Nature, that one, just showing that these cells are expressing a very important opioid receptor that are activated during the injury. And we really want to understand, okay, how does it work in mammals and how does it work in fish to try to see how we could activate this system maybe in humans. Right, it's a very big problem after injury. Yes?
Are you basically saying that as you age, this happens, and there's nothing you can do about it?
Okay, so the good news is that, no, the good news,
Repeat the question.
yeah, so the question is very important. It's are you saying that there is nothing to do about the aging process of, getting deformation of the spine? And so, to be honest with you, when I started, in 2011, there was no information whatsoever about why do we get deformation and whether it's a genetic disease. Now what we know is that there is clearly a genetic component. So if you see in your family that all people have scoliosis, you have good chance to have scoliosis, but it's multifactorial. So there is clearly multiple factors are going to give you a high risk to develop the deformation. But the beauty of it is that it's extremely plastic. So physical activity and all the activities that elongate your spine, really, swimming, or doing stretching, Pilates, I think everything you do in California is really good. So you have to stick to that. I think it's a yeah. In fact, I must say that when I saw you coming, I was like, oh, actually, you guys are much more healthy. No, because I think it's physical activity is essential, and it's keeping it throughout your life, trying to have as little injury as possible and doing the activities that elongate, always pushing, but, elongation of the spine is really, really important to keep it and to resist the fact that you could grow a little bit or degrow with time.
But didn't you say also pushing? You saying something about pushing?
Yeah, no, so it's, in fact, the dorsal root ganglia, they're sensing your pushing. So when you apply pressure and you actively push, I don't know if you've noticed that for the yoga in the room or people who do Pilates, you maybe notice that when you push, you actually get those spinal loop, sensory reflex, loop to be recruited, and it activates your abs and it recruits your perineum. So all your core is on, and that's really important because by doing that, you reduce the tension on your shoulder because you're really very strong in your abs, and you avoid to have this effect of oh, I have so many things to worry about. I don't know if you've noticed that. I notice that for myself. When I get very stressed, I go like this, So you kind of need to push, and doing many activities that put you in this position is really useful actually. Yeah.
Acupressure
Acupuncture?
Acupressure.
Oh yeah, acupressure. Yeah, so that one I haven't tried myself. You know, I've tried the other one I mentioned, but totally, yeah. No, in fact, working too much, I injured my back before 40 years old, and then I had like many exercises to actually like rebuild, and and I've noticed that all the exercise of strengthening when you're pushing are very, very important because you engage your core and deep muscles, but also indeed all the stretching, that's something I think where it's really complementary to what you do with running, right? Like you really need to go on those activities. Yes.
I wanted to ask more about the two-photon imaging. Can you talk a bit more about what exactly you're measuring and how it's providing the insights relative to your research?
Yes. So for us, it's been developed in Bell Labs with Webb and Denk, and that was before my time. But when I started as a grad student, that was like the big change in the field because suddenly, before two-photon microscopy, you only had visible light, visible lasers, and you were only reaching the surface of the brain. So you could only go to 100, 150 micron, and you were very fast, not able to go deeper in the tissue. So what they discovered in Bell Lab in the first place with Webb, and then Deng took it to the field of neuroscience, is that they could use infrared lights to have actually absorption of two photons with pulse laser. And by doing that, they could reduce the volume of excitation so that you don't bleach the sample. You go very deep because you have infrared light, and you get this beautiful resolution due to the fact that the two-photon effect is only localized to the focal point that you're scanning. So now we're using those technology. I didn't talk about it, but we use them to scan a sample very fast and image the activity in the brain. But we also use them now to activate the opsin I mentioned. So we come with a technology called digital holography, and we combine two-photon laser lights with...
Digital what?
So it's called digital holography.
Oh, okay.
And so we use spatial light modulator to shape light in the tissue. And then we use infrared light with pulse laser again, but at low repetition rate, to send a lot of light that we can shape in 3D like an hologram. And by doing that, it's on a different topic, more of motor control, but we can activate single neuron in the brain and activate hundreds of them in the brain. And so we're very excited because we start to see how many neurons do you need to do, the initiation of locomotion. And we start to put numbers on this, on how do you control the motor. And by looking at sensory and motor, we have hope to actually figure out the circuit, how it's organized to generate locomotion. And then our current project is actually to see in patients like Parkinson or in spinal cord injury, when you can't initiate locomotion anymore, could you actually use our knowledge from animals to target new areas in the brain, to stimulate with deep brain stimulation, an electrode, or maybe even with ultrasound, to do focal ultrasound and activate the brain completely, non-invasively in the human brain. Yeah.
Hi. Do you have information about the inner states in terms of emotions and like even the depression and other states? Because I know that affects posture, but also you can work on the posture to affect that.
There's a loop, there's a feedback loop, right?
Yes.
Yeah, exactly.
So
it's very hard to look at depression in a fish. But, right? I'm not gonna try. But the truth. No, but the truth is that I want to give an example, and maybe when I come next time to KITP, I can give you the result on that one. It's very interesting how wanting to put numbers in biology with my background, I actually realized that a lot of it is actually that you have your sensory inputs that can be positive, like, oh, I feel the sun, I'm feeling good, or it can be very aversive. There's a lot of sound, people did a party last night, I couldn't sleep, and you have those sensory inputs that actually change not only your motor output, but they also change your state, because what we find in the fish having this transparent brain is that they actually change the neuromodulation in your brain. So your serotonin, that is very important for depression, is immediately responding to sensory input. And same for noradrenaline that controls arousal. So we have in the lab a very cool piece of work that is on a different topic, but I want to share with you. You know, if you're under attack, and hopefully it didn't happen to many of you in the room, but if you're really in danger, the response that you're going to have that is conserved throughout the animal kingdom is actually to play dead. You know, it's a way for us humans to put sensor in sharks. We managed to put our hands on the electroreceptor with a glove protecting it. And when you put your hand on the electroreceptor that the shark used to find the seal in the water, the shark goes into a complete state of, they call it in the BBC channel or in YouTube, hypnosis. But in fact, it's not hypnosis, it's tonic immobility, where suddenly your body is completely, tonically stopped. Your heart rate goes down, and you're not going to have any more, ability to respond to sensory information. So it's like playing. It's a little bit different from a freeze. It's like playing dead. It's like more like the deer when it's attacked by the tiger, and you just play dead and pretend to be dead, hoping that the tiger will go, And we do that as well. It's a little bit sad to talk about this, but when you look at 90% of, victims of rape are actually playing dead, and they don't have any proof that they got, they were not approving. And that's a big problem for us in France, for instance, in terms of law, because you don't have ways to show. And so we find the mechanism is like aversive sensory cues are not only activating the motor response at first to escape or to struggle. At some point when it's an overload of sensory information, you activate the noradrenergic system, and it triggers a wave that stops completely the motor output. And the animal is completely, immobile and plays dead. And that is extremely conserved. We have evidence for it also in Drosophila. And we understand that it's basically sensory information that enable you, when you're not able to control and escape, that enable you to change state, actually, and enter into this tonic immobility state. And I see a little bit the same for, the circuits that control depression. We see that they're also sensing all the information coming in, either positive or negative, and they react to it, and then they will change plasticity in your brain. And that's why you get also this addiction to antidepressants, because they're super strong, and they change the connectivity in your brain. So we are really curious to look at those questions, but with a motor readout, but to understand, okay, how do we change motor states to be able to provide some guidance on how do we change mental states, We have this idea in mind that the way the system got built is to neuromodulate first the motor, because that was the first thing that animals do, is to control their locomotor output and their posture. And then the same neuromodulation from the raphe or from the locus ceruleus project into the brain to change the arousal and the attention in your brain.
So we do somatic work in therapy.
Oh, great.
Yeah, so grounding exercises, all that.
Oh, that's fantastic. Yeah. Oh, that's really cool. No, I'm very curious about that, because I don't study that, but I can totally see that it's so important. Yeah. And I do believe, honestly, that we need to develop a little bit on our own resilience and try actually to do a lot more with the plasticity of our brain. I feel like, and the plasticity of our body, to be honest. We have this amazing plasticity that remains at the adult and late stage. And if you use it, you can actually avoid a lot of the pharmacological help. Yeah.
Sounds like a great place to
Thank you. Thank you.